I'm titling this video Making Sense of the Chapter 7 Activity Table, or a walk through catabolism. In this video I'm going to walk you through the tables that were the activity for this chapter and kind of give you an idea of why I set them up the way that I did. So first, let's take a look at glycolysis. The substrate for glycolysis is glucose. Now, I confused a lot of you with this one because it's one glucose and I'm asking for substrates. But when you see the parentheses around here, you're supposed to read it either substrate, so one glucose, so that's the substrate per glucose for glycolysis. Or later on when we have two pyruvates, then you're supposed to read it substrates per glucose. See how that works? Anyway, glucose, through the process of glycolysis, becomes two pyruvates. Well, these two pyruvates become the substrates for the next catabolic pathway. Isn't that cool? So these two pyruvates become two acetyl-CoAs and two CO2s. And once again, these two acetyl-CoAs become the substrates for the next metabolic pathway. So let's take a look at the CO2. Heterotrophic cells cannot make use of CO2. You have to be an autotroph. So if you don't have the enzymes to deal with these CO2s, they diffuse out of the cell. And the same thing happens down here with the CO2s that are generated through the Krebs cycle. So they leave. So they do not become substrates for the next catabolic reaction. Now on to the electron transport chain. The substrates that are not involved in carrying energy around are the oxygen, hydrogen ions, worn out electrons, and water. So let's go ahead and skip down here to fermentation. To go on to acetyl-CoA and the Krebs cycle in the electron transport chain, you got to have oxygen. If you don't have oxygen, we stop here. And these two pyruvates go to fermentation if the organism has the enzymes to undergo fermentation. If I am a yeast, I'm going to produce two CO2, and once again, they diffuse out of the cell. I'm also going to produce two ethanol. Now, if I'm a lactic acid producing bacteria, I produce two lactic acid, etc., etc. Now, these also leave the cell. There are very few organisms that can take fermentation byproducts and make use of them later on. And that's my story, and I'm going to stick to it. So if you find out that that's not the case in later classes, realize I'm being overgeneral and being simplifying things for you. Now we're going to take a look at molecules that carry energy. By that I mean ATP, NADH, and FADH2, which are involved in catabolism. We're not going to worry about the other ones. Now, in going through this, it's good to know what they start out as. So, for example, the low energy form of ATP is ADP. The low energy form of NADH is NAD+, which I have down here because it's important in fermentation. And FADH2 is FAD. So keep that in mind. When it's produced, we're making this. When we are consuming it, we're making NAD+ out of this. Now over here, I have where these various pathways take place in the cell, but I'm going to talk about that later. Let's talk about these energy molecules. So the ATPs produced in glycolysis and the Krebs cycle can go on to anabolic pathways and be used immediately. So they can be used to make proteins from amino acids. Remember, amino acids are smaller than proteins, so that's an anabolic process. We can make DNA and RNA from nucleotides. Once again, nucleotides are smaller. You make a bigger molecule. So ATP is being used for this. But what about this NADH? Two of them that were produced during glycolysis, and two NADHs from acetyl-CoA. So let's go ahead and circle these. And we've got some NADH and FADH2s. These need to be converted to ATP before they can be used in anabolic processes. So we send them to the electron transport chain. And through the electron transport chain, we make ATP out of it. Now, I will refer you to the textbook for details on how the electron transport chain does that, but let's go over the numbers. Approximately per NADH, I make 3 ATP. And approximately per FADH2, I make 2 ATPs. So up here in glycolysis, if I have oxygen present, these NADHs, I can make 6 ATPs out of it. Here, from the formation of acetyl-CoA, once again, if I have oxygen, these NADHs can go to the electron transport chain, and I can make 6 ATPs. Now, for the NADH from the Krebs cycle, once again, you have to have oxygen, I make 18 ATPs, and I can make 4 ATPs from the FADHs. So I got these numbers because 6 times 3 is 18, and 2 times 2 is 4, so I get 24. So I add up these 2 net ATP here, these 6 ATP, these 6 ATPs, and then these 24 plus these 2, and that's where I come up with 38 ATPs for aerobic respiration. Now that's in prokaryotes. In eukaryotes, we have to spend some NADHs to get pyruvate from the eukaryotic cytoplasm into the mitochondria. So that's why we get 2 fewer ATP than prokaryotes, but that's still not bad. Now let's look at what happens when there's no oxygen. If there's no oxygen, these NADHs are not going to be sent to the electron transport chain. But I can't store these NADHs. They have a very short expiration date. They've either got to be used or I just let them lose their energy. Now, to keep running glycolysis, I need NAD pluses to keep glycolysis going so I can keep producing 2 net ATPs. So what happens is, during fermentation, these NADHs that are produced in glycolysis are consumed. So NAD pluses go back up to glycolysis because in fermentation we get rid of that hydrogen, and then the NAD pluses, the NADHs that are formed during glycolysis get sent back down to fermentation. They get turned into NAD pluses. They go back up, and we're able to keep glycolysis going. So the entire purpose for fermentation is to produce NAD pluses so that we can keep glycolysis going and we can still continue to get 2 net ATP to make proteins and amino acids and nucleic acids and all sorts of good stuff. Because 2 ATPs are better than no ATPs. Now you may be thinking that the purpose of fermentation from a human standpoint is to get some of the byproducts of fermentation, such as CO2 and ethanol if we want to make bread or wine or beer, or to make lactic acid so that we can get yogurt and cheese. But that's not the point for the cell. That is the point from the human point of view. So I want you to think about why the cell is doing it, to produce NAD+. Let's take a look at where all of these catabolic pathways are happening inside of a cell. So here's my cartoon drawing of a eukaryotic cell. See, I've got my nucleus up here. And here's my cartoon drawing of a bacterium, of an aerobic, respiring bacterium. So let's go over endosymbiosis, which is the theory that at one time we had eukaryotic cells that did not have mitochondria or chloroplasts yet, and they were anaerobic, were not capable of undergoing aerobic respiration. So at some point, an aerobic bacterium got inside the cell and was encased in a vacuole. See, here's the blue for the eukaryotic cell membrane. And because the peptidoglycan cell wall wasn't necessary, eventually we lost it. So here is a mitochondria that has an outer membrane and an inner membrane, or cristae. Now let's look at where the various catabolic reactions, where the various catabolic pathways happen within a eukaryotic cell versus a prokaryotic aerobic cell. So let's start with glycolysis. Remember, glycolysis does not require oxygen, and it takes place in the cytoplasm of our prokaryote. It also takes place in the cytoplasm of our eukaryote. From this, we can derive that both the ancestral eukaryotic cell, that had a nucleus but no mitochondria yet, had the enzymes to undergo glycolysis, as did this prokaryotic cell. Now let's go to fermentation. Fermentation also does not require oxygen, and that takes place in the cytoplasm of the prokaryote and in the cytoplasm of the eukaryote. Once again, both of these had the enzymes to undergo fermentation before endosymbiosis took place. Now let's move to the pathways that do require oxygen, starting out with acetyl-CoA formation. In aerobic bacteria, acetyl-CoA formation happens in the cytoplasm. But in eukaryotic cells, it happens in the matrix of the mitochondria. You could say it was happening in the cytoplasm, but we named the structures of mitochondria and chloroplasts before we knew or had the theory of endosymbiosis. But you can see that the aerobic bacterium that was engulfed by the eukaryotic cell maintained the enzymes necessary for acetyl-CoA formation. Now onto the Krebs cycle. This also takes place in the cytoplasm of the prokaryote, and it takes place in the matrix, or the cytoplasm, of the mitochondria. Once again, this ancestral prokaryotic cell brought the enzymes necessary for this metabolic pathway into the eukaryotic cell. Now, electron transport chains. In aerobic bacteria that are capable of aerobic respiration, enzymes are embedded in the inner cell membrane, and this cell membrane is folded into many folds so that you have more surface area so that you can have more of these enzymes. Well, the electron transport chain happens in the folded inner membrane of a mitochondrium, or the cristae, so it's happening in about the same place. All of this supports the theory of endosymbiosis, and it also helps us explain what's going on where. So hopefully this walkthrough has been helpful, and as always, if you have further questions, feel free to ask me. That's what I'm here for.